I was under the impression for many, many years that cancer was a genetic disease. There's no question about it. I was indoctrinated just like almost everybody else.
So you actually kind of talk about some of this in your recent paper, which I've actually sort of printed out. It's very provocative.
Once you realize that it's a mitochondrial metabolic disease, I think we can drop the death rate by 50% in 5 to 10 years, depending on how you go after it. You're going to see that needle drop.
In your experience, does that kill cancer cells if you remove the sugar?
I don't believe so.
Mitochondrial inefficiency is necessary and sufficient. I think we always have to put into perspective.
Though, is a known risk factor, right? So
well, you have risk factors for sure. But is it a primary risk factor or a secondary? Yeah, well, thank you again, Lauren. Thank you for bringing us together to discuss this. I'm Tom Seyfried. I'm professor of biology here at Boston College, and I teach cancer metabolism and to the seniors, mostly juniors and seniors and grad students, and also general biology to the non-majors. And I do spend considerable amount of time discussing cancer, even because I find that cancer is a field that can interact and integrate many aspects of biology and cell biology, biochemistry, and genetics. And if you try to give those subjects in a very dry way, it goes in one ear and out the other. They don't really assimilate. But when you link it to a particular significant problem like cancer, then they can begin to tune in a little bit more to the importance and make these biological linkages in relationship to a disease. So we can touch upon a lot of different parts of cell biology, genetics, metabolism in a general biology class just by integrating the concept of cancer and how it affects us. So this is what I do, and a week from today, I will start my classes here at BC, and we'll run for the whole semester and next year as well.
Sounds good. Yeah, and I've had you on the platform before to talk about what you have seen in research, and it definitely goes against what I would say that the typical understanding of what these cancer causes are. I'm going to ask you the same thing, Dr. Bunz. What a little bit about you and what you're bringing to the table as far as understanding cancer.
Yeah, sure. So I'm Fred Bunz. I'm an associate professor of radiation oncology and molecular radiation sciences at Johns Hopkins University. I'm also a member of the Kimmel Cancer Center there, which is a comprehensive cancer center that does research, training, and sees people with all kinds of cancers. So my primary role at Hopkins is as an investigator. So my lab tackles sort of a variety of subjects that mostly revolve around one particular tumor suppressor gene, a gene that's involved in cancer called P53. We also work on some cancer therapeutics and gene therapies in my lab. So the other major hat I wear is as an educator. So I'm one of the directors of our program in cellular and molecular medicine, which is like the largest graduate program at Johns Hopkins School of Medicine. So it's basically, as it sounds, it's a PhD program. So I do quite a bit of teaching in that. I run a number of summer programs at our cancer center, including a program where we bring medical students from around the country to hopefully get them excited about cancer research and hopefully have them consider careers in oncology. And I guess, and thank you, Lauren, for this invitation. And I guess I'm really here to sort of represent the consensus view that cancer is a genetic disease. And I'm sort of looking forward to discussing, why I think that, and why, and probably less important what I think, but, why is that the consensus view? Where does that come from?
Right. Awesome. Yeah. Thank you. And definitely worth noting too, the way I found you was through your work. The book that I researched that came up was called The Principle of Cancer Genetics. And that is a book widely used. It's a textbook more or less used by tons of colleges and in different courses to educate people on cancer. So that was your book. And that was one of the biggest one of the most reputable books I found when doing this research for who to bring on and talk with Professor Seyfried. So really, really two very impactful people here. I think to simplify all of this, like you said, Dr. Buns, you approach this from seeing cancer as a genetic disease. And then Professor Seyfried, you kind of approach this from more of the Otto Warburg theory, which opposes that view. And we're going to get into all of that here in a second. So the way this is going to go is obviously just a conversation that we're going to have, but I'm going to have some questions that I'm going to go through, and I'm going to, list off just one by one. And obviously you guys will just get a couple minutes to respond, and then, state your opinion or your facts that you have. And then we can go back and forth for each person to have a chance to respond and toss it around. So without any further ado, I'm going to start with my first question here. And thank you both for being willing to do this. And I'm excited. I'm excited to get into this. So the first question that I have is really something that's been very interesting to me now that I've been living life eating mostly meat. I do mostly a carnivore diet, carnivore lifestyle, and I have seen how much that's benefited me in my health, in my mental health, physical health, everything. So, from my understanding, two of the bigger factors that go into causing cancer, as far as, what could be part of the somatic theory, kind of like what you're representing, Dr. Buns, is one, smoking is a huge cancer cause, it seems like, and two is obesity and the amount of body fat that one actually has, on their body. Those are two very large arguments as to what can cause cancer. There's an argument to be made that body fat, that the higher amount of body fat could be causing it. But that also that might have nothing to do with it. Actually, it might be, in fact, an issue with what might be causing obesity. So that could be, for example, insulin resistance or chronic inflammation. Those are two things that come with obesity. So it's not necessarily obesity that's causing cancer, but it's in fact the insulin resistance and possibly inflammation. Is it possible that cancer is caused by insulin resistance and inflammation? And I'm going to toss this first to you, Professor Seyfried, if you wouldn't mind addressing that.
Yeah, well, let me just say, first of all, that as we know, right now, cancer is killing about almost 1,700 people a day in the United States from the work of the American Cancer Society, which comes to about 70 people an hour. And these dead bodies pile up year in and year out. So obviously, we have a major problem that seems to be getting worse year in and year out. We're not making any major drop in death rates, or I would say death rates in dead bodies per day. So, but as far as the obesity epidemic is concerned, obesity is an effect of an imbalance of nutrients. There is a correlation between obesity and risk of cancer, and some have said that by looking you see more cancer in obese people, but I think it is a reflection of the inflammation and insulin insensitivity, and you have a lot of underlying metabolic problems that go into this. So the question is, how would systemic inflammation in the body lead to a tumor in a particular organ of a person that would have this? And the answer is, we and others have shown that obesity, that inflammation can damage morphogenetic fields and mitochondrial function. It creates oxidative stress in the mitochondria of cells in a particular organ, and this leads to mutations in the nucleus and dysregulated cell growth. So inflammation is a provocative agent that can underlie a number of different disorders, cancer just being one of them. But how we view inflammation as the linkage to the damage to the respiratory system in the cell, producing reactive oxygen species, which then lead to nuclear gene mutations as a downstream epiphenomenon. But the issue, of course, is dysregulated cell growth. how does that lead to a cell going out of control? And when a cell has damaged oxidative phosphorylation, it falls back on the default state of fermentation, and fermentation is linked to dysregulated cell growth. So we've put together the pathway from provocative agents like inflammation or these kinds of things, and specifically linked to obesity, because people do understand that there is some sort of a connection between the obesity, but it's more linked to systemic inflammation. And also, you have elevated glucose, which drives the dysregulated fermentation of these cells. So, blood sugar is there, elevated, but that's just a facilitator, it's just the fuel. What's causing the problem is the damage to oxidative phosphorylation, leading to secondary downstream effects. That's the way I would address that question.
Okay. And I just really quick before I get to you, Dr. Buns, I want to clarify. I have spoken with you, Professor Seyfried, before about this, and I had to get like a very much clear on what you're talking about. There's a lot of science going on in there that I don't necessarily know about, right? So for people that are listening, and tell me where I'm wrong with this, but I'm going to try to get it right. Ultimately, cells are supposed to use oxygen to generate energy. And what happens in your theory is that, once a cell can no longer use oxygen anymore, they have to revert to, basically old school ways of generating energy that is something only like cancer cells really do. Normal cells generate energy through oxygen. And so basically the issue right now is that you're saying many of these cancer cells in your theory cannot produce energy correctly. And so they have to fall back into, what's it called?
Fermentation.
Fermentation.
It's energy without oxygen.
Energy without oxygen.
So yeah, and all cancers that we know of have a dependency on fermentation. In every organ, every type of cancer cell, they have a dependency on fermentation, and that's when you look under the electron microscope and you look at the number, structure, and function of mitochondria, and you see in every major cancer all major cancers, and I published this, they have abnormalities in the number, structure, and function of mitochondria. But it doesn't happen overnight. It's a protracted problem. It can take months, years for the cell to transition away from oxidative phosphorylation to substrate level phosphorylation, which is the origin of fermentation. It doesn't mean that there's no oxidative phosphorylation. It just means that it's insufficient. The cell cannot survive. It cannot live without energy. Energy becomes the central issue. And people need to know how every cell in our body has to have energy. Where does the energy come from? Yes, we breathe air. The oxygen drives energy in our cells, but cancer cells can can survive without oxygen, and even when oxygen is present, they continue to ferment. They continue to ferment because the oxidative phosphorylation system is insufficient. It's not gone completely; it's just insufficient. So you have to have a sufficient level of energy. And don't forget, the singular largest consumer of energy in any given cell is the membrane pump. 30 to 70% of energy comes from the pumps. To maintain an electrical potential across the membrane. And if that pumps don't get energy, the cell swells and dies. It undergoes death. And if you stop oxidative phosphorylation, like cyanide, it will kill you quickly. But cancer cells can live in cyanide. So clearly they're not using oxidative phosphorylation. They're fermenting. And that's the singular biggest difference between normal cells and cancer cells. Cancer cells can live and grow without oxygen and ferment.
Yeah. So, okay. So basically you're saying yes, that inflammation and all of the insulin resistance can absolutely be a cause of this cancer. Now I want to move this to you, Dr. Buns. What is your thought on that question? I'll restate it. Is it possible that cancer is caused by insulin resistance and inflammation, and it's not necessarily the smoking or the obesity that is causing it? If that makes sense.
Yeah. So let me just start very quickly I just want to address one point that Professor Seyfried mentioned, which is the sort of the rates of cancer incidence. So I think it's an important, and this is a thought that gets bounced around like, oh my gosh, there's more cancer than ever. In fact, the incidence rates of most common forms of cancer have been steadily dropping for the last couple of decades. colon cancer, breast cancer, um, and stomach cancer, probably most is one of the ones that's really gone from a major source of cancer morbidity, mortality to one now that's really one of the more rare cancers. So in general, the news is quite good. Now, Professor Seyfried is quite correct in that the absolute number of people that are dying from cancer is still high. It's way too high. However, part of that is that we're not dying from other causes as much. People are living longer. Our population is aging. It's one reason that cancer centers like ours are pretty much have all the business we can handle. But the good news is that for most common types of cancer, the incidence is going down. Now, unfortunately, that's not true across the board. Pancreatic cancer, which is one of the most lethal kinds of cancer, it's very stubbornly stable in terms of incidence. And in fact, we're seeing while the overall rates of colorectal cancer are going down precipitously in recent years, a really sort of distressing finding is now that more younger people are getting colorectal cancer. Whereas largely, cancer by and large is a disease of aging. So there's definitely something that we're still trying to figure out as to why that is. But again, the news is good. The other key point I want to make too is survival after cancer diagnosis is now way better than it's been. The dramatic changes since the 1970s, when the current regimens of radiotherapy, chemotherapy, and more recently targeted therapies have come online. The results have been really strikingly positive. People are living longer with cancer and with longer time, until recurrence. So, which is really, real life extensions with some of the therapies that are currently being used. So anyway, I didn't want your listeners to,
Sure.
I want you. I think it's an important thing to be aware of. So it's not all gloom and doom, but yes, we absolutely need to do better. So getting to your question, so inflammation is unquestionably a critical aspect of tumor growth, and that is very clear in experimental models. It's very clear when we actually look at cancers under the microscope. We can actually see the inflammation. We can see these inflammatory infiltrates. So cancer, inflammation is both part of the tumor itself. So it's what we call in the tumor microenvironment. There's sort of a whole inflammatory aspect for most sort of common solid tumors. And furthermore, sort of systemic inflammation is clearly a driver of cancer. Now, maybe there's no tissue where that's sort of more vividly evident than in the colon. So there's a fairly common disease called ulcerative colitis. So it's basically an inflammatory disease that mainly affects the colorectal epithelium, and it's basically sort of chronic inflammation in the colon. And it's well known that patients with that disorder have a greatly elevated risk of developing colorectal cancer later in their lives. So again, that's just one of many examples that show the concrete, relationship between inflammation, immune dysfunction, and cancer. So in that regard, I will agree with Professor Seyfried, regarding the importance of both inflammation and also metabolism. One thing I want to touch upon, I think, Lauren, that you mentioned was this idea that sort of the Warburgian metabolism, the famous Warburg effect, and somehow the cancer gene theory are somehow in opposition. Really not true at all. I mean, it's actually the Warburg effect is real. It's pretty much universally accepted among people in the cancer research field that the cancers, as Professor Seyfried has mentioned, are they they basically function metabolically in this mode that we call aerobic glycolysis. So that sort of fermentation in the presence of oxygen so it's a very unusual type of metabolism that is interestingly also a characteristic of pathogen infection.
Mhm.
So it's definitely part of the disease process. But as I'll describe, as we go on to discuss, it's actually not the underlying cause of the cancers.
Okay. And inflammation is, or sorry, insulin resistance, is that all coupled into your response there too, or is that just with inflammation?
Yeah, no, it's really they're
Okay.
very interrelated. So
Okay.
it's very well known that in a sense obesity does have inflammatory aspects. It's known that people that are obese have issues with inflammation in various tissues. So they're really some of these systemic. That you could talk about local inflammation, like in the case of ulcerative colitis, but it's also sort of systemic, inflammation that it has been associated with obesity. And in both of those
Mhm.
cases, it's definitely an important ingredient in sort of promoter of sort of tumor progression.
Okay. Okay.
Um,
Go
Lauren,
ahead.
can I ask Fred a question?
Yeah, please.
Sure. I appreciate your looking at cancer as the rate, but when President Nixon in 1971 declared the war on cancer, when we talk about wars, we don't talk about the number of soldiers killed per number of people in the society. We count dead bodies. That's the way we talk about war. And when you have 1,700 people a day dying, I don't know how anyone could sugarcoat that number. This is a 70 an hour. I mean, we can talk about the rates and we can talk about this. You got 70 souls an hour dying from this disease, 1,700 a day. And then when you look at that and you say they live longer, many of these poor folks are suffering miserably from the toxic therapies that they were given to give them an extra overall survival. And many of these guys, they don't live very much longer. I work in glioblastoma. They haven't made a singular advance in 100 years, 100 years in glioblastoma. And you get metastatic cancer, and you're just as going to, well, you have your survival is no different today than it was 75 years ago. If you treat stage 1 tumors, you're going to have a lot of success, and you're going to keep people alive a lot longer. But let's not try to sugarcoat 1,700 people a day dying from cancer. I don't think that's good news to anybody.
Let me be very clear. I am not interested in like sugarcoating, that the death toll. Again, it's really, a leading cause of death in our society. So there's no way around that. However, I think our audience can take some perhaps solace in that your risk of cancer as an individual is going to be lower than it was for your grandparents. And that's where the number is sort of, I think the incidence rates become very significant. So again, there are more people. Yeah, we have a larger country than we did 20 years ago. So all these, there are many aspects, ways that you can look at it. So, I guess, maybe, I'm more of an optimist. I'm very excited by the progress we've made, but I'm also motivated to sort of continue the progress into tumors like glioblastomas, where I totally agree. There's been very, very little progress in those types of tumors. But I think rather than focus on those, we could all at the same time say, well, the other side of the coin is there's some cancers that were almost universally fatal. Chronic myelogenous leukemia, for one, which now is a very survivable form of cancer. So we're living in a world where cancers that were killing people a generation or two ago are now very manageable.
Dr. Seyfried, would you agree with that?
To an extent I would, but I still think we're not making any major progress. The death keeps going up. I count dead bodies, and every year they get worse and worse. If you have something that works, like the protease cocktail for AIDS, you got a dramatic drop in death. It was like overnight you could see. We have continual increase in cancer. yeah, we're starting to treat a lot of cancers far earlier. These zero cancers, stage ones, and these kind. You can have success with that. However, if you're going to radiate and poison people to make them healthy, you also put them at risk for later on afflictions. And I see so many people coming. They do live longer, but they're living longer from a minor tumor. You take a guy with stage 4 lung cancer, he ain't going to live that long. And the treatments they give these guys today are just horrific. So I'm looking at a situation that's out of control. I'm not looking at something that we're making any major progress. And I'll let the people judge for themselves. All you have to do is look at the obituary pages, and you're going to see the numbers of dead people, politicians, movie stars, businessmen, industrialists. These guys, we're all at risk. And yes, they're younger. The people, I agree with Fred. There's no question about it. I'm seeing it myself. We're younger and younger. I can't believe how many emails I'm getting from people in their early 30s and early 40s with colon cancer and all these kinds of cancers, and it basically has to do with our diet and lifestyle. We're eating more highly processed foods. We're not exercising nearly enough, and we're putting ourselves at risk not only for cancer but for a variety of other chronic diseases as well. But I was under the impression for many years that cancer was a genetic disease. There's no question about it. I was indoctrinated just like almost everybody else. But then I started to see cancers that have no mutations, and I said, how do you explain cancer as a genetic disease when you have some cancers that have no mutations, as Stratton's group and Parsons' group and Verteg's group and all these groups are seeing no mutations in cancers. I find that shocking. And then I thought, well, maybe there's only some mutations that lead to this, like these driver mutations. And now we find all these driver mutations in normal tissues that never become cancer. I said, how is that possible? What's going on with that? And then the nail in the coffin to me was the nuclear transfer, mitochondrial transfer experiments. I mean, when you view all of these inconsistencies together, you can't rationally think that this is a disorder of genetic regulation. We have never found a tumor that goes without fermentation. We have never found a tumor with normal number, structure, and function of mitochondria, regardless of what their genetic mutations are. And Fred knows that almost every cell in a tumor has a different genetic profile than any other cell in the tumor. Different tumors, there's thousands and thousands of mutations now that are being found in these cancers, but they're all fermenters. So the common phenotype of cancer is fermentation metabolism driven by insufficient oxidative phosphorylation. We have found no tumors that are different from that, although they are different genetically for sure.
Okay.
How would you, Fred, tell me how you deal with tumors that have no mutations?
Yeah. So,
explanation?
that's an interesting question. So I was looking at, so you actually kind of talk about some of this in your recent paper, which I've actually sort of printed out. Is very provocative. I guess the simplest way to address that is there are really virtually no tumors that have been detected with no mutations. Okay, so the papers that you cite in this manuscript, so the one from the Stratton group, which is a well-known, it's basically the Sanger Center in the UK, which is a major sequencing hub globally. So they did a study in early 2007. Where they basically looked at a panel of, I think, 550 or so genes, and they were able to use sequencing technologies at the time that were very novel and powerful, and they found mutations in many of those tumors. And so the genes that they chose to look at were genes that encoded a class of protein that there were enzymes called protein kinases. They were known to play an important role in signal transduction, and notably, protein kinases are actually enzymes that we can actually target with some drugs. So they took a very focused look at this set of genes to look very broadly in a series of several hundred cancers to see whether they would find mutations. Okay,
Well,
so
what did they say about the 73 tumors that had absolutely no mutations?
that's in 550 genes. Okay, we have about 20,000 genes.
No, but
So
they had no mutations of the 550 genes in 73 out of the 200 tumors they looked at.
right, yeah, and
So
that's fine.
that's tumors without mutations.
No, they didn't sequence the rest of the genome. They're looking at, what is
Okay,
that?
so they did not do a deep dive on these tumors then. They found
No,
some.
so they didn't have the ability to. So notably, and this is maybe a little plug for my institution, later that same year in 2007, the very first complete cancer exomes were sequenced at Johns Hopkins University. So in those cases, they were actually able to look at all the 20-something thousand protein encoding genes, and what they found mutations in all of them. There were no tumors where there were
Okay,
no
well,
mutations.
I just saw the 73 that didn't have. Well, and the new work by Verteeg in 2020 says that we can't find mutations in ependymoblastomas. So it's not just some. Yes, I agree with you 100%. These are not common. But if you find any tumor where there's no mutation, like the B20 tumor, that the glioblastoma had no mutations in any of the three major signaling cascades, it raises a question. Like, okay, and when I looked at the papers that had these no mutation, the authors of those studies never addressed that. Why would you not ignore an observation not consistent with your hypothesis?
Yes, so
Even
I
if you
think
didn't finish the deep sequencing.
misunderstanding. I think you're misunderstanding the data with all due respect. So again, that the paper by Parsons et al. You just mentioned, looking at the brain tumors. Again, that was another study done by my colleagues here at Johns Hopkins. You're right; they found no mutations in these three major signaling pathways, but they found other mutations. Again, remember, this is a time of discovery when they're discovering more different pathways. So IDH1, right, a gene I'm sure you're very familiar with. Frequently mutated in gliomas was not known about then. It was only subsequently, delving deeper into the genomes of these tumors and finding these recurrent mutations. And by recurrent, Lauren, I mean they find them in patient after patient. So not 100% of patients will have every characteristic mutation, but all tumors have some mutations. Now,
Well, I mean,
No,
we can debate that based on the data. I would have the people look themselves at the value, have them come to their own. I looked at it and I saw tumors with no mutations. But I have to be honest with you. Yes, I agree. Most cancers have a lot of mutations, and not every cell in the tumor has a different mutation. Some are common, like p53, but not all tumor cells have p53 mutations, as you know. But how do you explain driver gene mutations now in all these normal tissues that don't get cancer?
So something I want to bring us back to, because we can go on in all the weeds. That's the thing. You guys are such experts in this that you can go in, and I want you to. But I also want to bring it back to the level that person like me or somebody else who has cancer that doesn't really know much about cancer can understand. And one of those questions, like, what I think is really important is to really, in just one sentence, say, if you can, and I know like it might be a long sentence, but like a very simple sentence, what do you believe causes cancer or could be causing it? Because I think we need to clarify what Otto Warburg thought in simpleton words, right? And then also clarify what the genetic mutation theory is, what I guess it would be the somatic mutations theory is. And then I'm going to go into my next question. So in one sentence, Dr. Bunz, can you say what the somatic mutations theory is?
well, so at the outset, let me just mention, I prefer the cancer gene theory as the way to sort of describe it. And the reason for it is that not all cancer-related mutations are somatic, meaning they don't all occur during the life of the individual. That in fact, some of them can be inherited through our germline, either through parents or during de novo mutations that occur during embryonic development. Again, I don't want to go too deeply into the woods, but so essentially, what is the cancer gene theory? So the cancer gene theory, sometimes referred to as a somatic mutation theory, basically holds that cancer is a result of the accumulation of critical mutations in genes that control cell growth, cell differentiation, and the ability of cells to be recognized by the immune system. Um, and that is and it's really the consequences of those mutations are what give rise to all the different what we call cancer cell phenotypes, including the Warburg
Okay.
effect, including aneuploidy, which is having sort of a you know a non-modal number of chromosomes, a whole bunch of different characteristics of cancer cells, are basically are all things that we need to know about to understand the biology. But again, the underlying cause of those effects are genetic mutations.
Got it. Okay. And yes or no, are they some of them are inherited, not all of them, right? The mutations.
Correct. Most of them arise during the lifetime of the individual, but in a small number of cases, but significant number of cases, cancer mutations can be inherited.
Okay. And so what you're expressing there is the generally adopted theory behind cancer. That's, I would say, the theory that I would say most people would agree with. Now, obviously, Professor Seyfried, you're of the opposing side to that. Now, poke holes in what is your theory? Please explain what the Otto Warburg theory is, and then what's the difference between that and what Dr. Buns just explained?
Yeah, well, Warburg claimed that all cancers were fermenters because the oxidative phosphorylation system was inefficient and they had to ferment. And we know that fermentation is the default energy state. This is the state of all organisms on the planet before oxygen came into the atmosphere 2.5 billion years ago. So at that time, all the cells from fossil work, cellular fossil work, were their dysregulated cell growth driven by a fermentation metabolism. When you develop cancer today, the cells get abnormal energy metabolism. The oxidative phosphorylation becomes corrupted. And that leads to the formation of reactive oxygen species, which are carcinogenic and mutagenic. And Dr. Buns has gone through knows that ROS, or DNA damaging reactive oxygen, the nitrogen radical, the oxygen radical, they will cause mutations in the nuclear genome, in the DNA. It's known ROS cause mutations. They come from the mitochondria. Why? Because the oxidative phosphorylation system is corrupted. It's inefficient. And if it's inefficient, the cell has to turn on fermentation. And we see that all cancer cells are fermenters. We have never found a cancer cell with a normal oxidative phosphorylation system. But the other issue, of course, is when we take the nucleus out of it, all of the nuclear mitochondrial transfer experiments were very revealing in letting us know whether it was the genes in the nucleus or it was something in the cytoplasm that was responsible for the dysregulated cell growth. I think that binging is coming because I'm using the Chrome. I don't usually use Chrome, and I think something is strange with my computer on the Chrome. But that's a different issue. But let's look at this. When you take the nucleus of a tumor cell with the somatic mutations, with the driver mutations, and you put it into a normal cytoplasm, you get regulated growth. You don't get dysregulated growth. So if these genes are causing dysregulated growth, why you don't get dysregulated growth when you transplant the nucleus from the tumor cell into the normal cytoplasm? And if you do the reverse experiment, that is, if you take the normal nucleus, which has normal integrated genome, and put it into the cytoplasm of a tumor cell, you get dysregulated growth, which is cancer. And this has been done in multiple in vivo and in vitro experiments.
So the thing that I want to understand.
Well, I would say that my question was, if this were a genetic disease, you would expect the nucleus from the tumor cell to derive dysregulated growth, which is cancer. And it doesn't. But if I take the normal nucleus and put it into a tumor cytoplasm, I get dysregulated growth. So that clearly tells us it's the cytoplasm. And in the cytoplasm, you have mitochondria, and the mitochondria energy dictates the cell cycle in the cyclins. through the electrochemical gradient in the inner membrane of the mitochondria. The mitochondria is responsible for cancer. Damage to oxidative and the mutations in the nucleus are effects of the ROS produced in the mitochondria. So
Okay.
I'm looking at some cancers with no mutations, driver gene mutations in normal cells that never develop dysplastic growth, and I'm looking at the nuclear transfer experiments. Any one of those factors alone would raise serious questions as to the validity of the somatic mutation theory.
Okay, so with that said, and I know I know you want to respond, Dr. Buns. Just to yeah. So to keep us on track here, something that I'm very curious about is understanding the impact of sugar on our cells and how that might cause issues with, let's say, mutations, or let's say the mitochondria. Right? Is there some kind of understanding that we can we agree that sugar is a problem when it comes, like, overconsumption of sugar is a problem, even if it's just two different theories here? Dr. Buns, would you agree that sugar is an issue, sugar consumption, and that we could probably tie back into insulin resistance, as a result?
Yeah, to some extent. I think access to large amounts of readily digestible carbohydrates like sugars, is a well-known factor that can promote insulin-resistant diabetes later in life, which is absolutely a risk factor for cancers. So I think
Okay.
diet is a very important aspect of what we call, we all have what we call risk factors, right? Some of them are not modifiable, meaning there's nothing we can do about our parents, there's nothing we can do about our genetic inheritance, and there's nothing we can do about random mutations that arise during cell division. However, we can control our behaviors like smoking, alcohol consumption, and certainly diet is another highly modifiable risk factor that we can really consider when we talk about cancer prevention. Not that it's easy to, for people to actually, change their lifestyle and change their diets. But many people would stand to benefit by taking a hard look at what they're eating.
Okay, and with your theory, would you argue that if the way that you're saying that, how you understand the theory is that you might just have the genetics that they become mutated, and sugar might just not help that? It's not going to be helpful, but it's not necessarily sugar that's the cause, right?
Well,
Or like diet or something like
yeah,
that.
yeah. It's so we really talk in terms of risk. So again, insulin dependent, insulin resistant diabetes is certainly a cancer risk factor. Not all people, with that disorder are going to develop cancer. So again, it's a numbers game. We see a disproportionate number of people that have sort of these metabolic health issues. For sure, and it's hard to deny that is likely playing a causative role. And many people are looking at, perhaps that's why we're seeing increasing rates of cancer in younger people. Now, no, those increases are very, as I mentioned, very concerning, but they still, as a whole, represent a very small proportion of cancer overall. But obviously, when cancer is occurring in younger people, we really stand up and take notice. So there are a lot of people now taking a really sort of deep dive into what might be going on in terms of our current modern day exposures, diets, and I think again, diet, obesity, and hormone exposures are really, I think, at the top of many people's lists as they think about, what might be causing these cancers in young people.
Okay, and removing sugar, in your opinion, and I'm specifically sitting on the sugar thing, I know that I'm kind of harping on it. In your experience, does that kill cancer cells
I
if you
don't
remove the sugar?
I don't believe so. Because the body has a lot of ways of producing those, the fundamental building blocks of metabolism. So you don't necessarily have to have processed sugar. So my short answer to that is no. But I think, all things in moderation is sort of generally, a good a good way to kind of consider your own diet.
Got it. Okay. Thank you. Yeah. So let's go to you, Professor Seyfried. I'm going to ask you the same question. Do you think that sugar has could sugar potentially be a cause of cancer issues that you're seeing specifically in the mitochondria and generating energy?
Well, sugar, let's be honest, sugar is not a carcinogen. You can use the Ames tests and put sugar on. Sugar does not cause mutations in the nucleus. Sugar itself does not damage mitochondria. What damages mitochondria is systemic inflammation, radiation, intermittent hypoxia, oncogenic viruses, all of which impact negatively on the structure and function of the mitochondria. If you have a neoplastic growth in your body, elevated sugar in the bloodstream will make the tumor grow faster because it's a prime fermentable fuel. But the sugar itself is not a carcinogen.
Mhm.
So you have to separate causality from what's actually driving the growth of the cell. Yes, if the numbers of studies that have been shown in people with a broad range of cancers, whether it's breast, colon, brain, lung, liver, if you have high blood sugar, those folks don't live as long on the planet as people that have lower levels of sugar, glucose. Let's put it this way: it's glucose. Don't forget, we evolved in a Paleolithic period for tens of thousands of years, where highly processed carbohydrates were not present anywhere in our diet. If you Albert Schweitzer looked at native populations that lived according to their traditional ways, cancer was extremely rare in these folks. In fact, he looked at 40,000 people in Africa and find one. It's in the book, Noakes's book. The Inuits of the North regions, their cancer was unknown. As soon as they started the Western diet and lifestyle, little exercise, large amounts of highly processed foods, they got systemic inflammation, obesity, chronic heart disease, cancer, and a variety of other disorders. So sugar itself is not a cause of cancer, but sugar and all the other highly processed foods with little exercise. Don't forget, we evolved as a very active species. We were not sitting around getting hamburgers and donuts through a window at a fast food shop. We're putting ourselves at risk for cancer and a variety of other chronic diseases because of our diet and our current diet and lifestyle. Our technology has evolved so much faster than our biology. And as the result of that, we put ourselves at tremendous risk for these chronic diseases, cancer being a major one. So,
Okay.
yeah, I wouldn't get too hung up on sugar. I would get hung up. I would be more concerned with your general diet and lifestyle. And this is why we built the glucose ketone index calculator to allow people to know if they're in nutritional ketosis. Don't forget, we evolved in nutritional ketosis. That's who we are as a species. We would have low blood sugar and elevated ketones because there was very little carbohydrate in our diet. We killed and ate animals. We ate
Let me
fish.
ask you this, though, with that. Let me ask you this on that point, because do you think if one were to remove mostly sugar from their diet, it would then kill cancer cells? Let's say, combined with other things too.
Well,
Same question I asked Dr.
Cancer
Bunce.
can't live without the glucose and the glutamine, the amino acid glutamine. So if you're going and you have to transition the whole body over to ketones and fatty acids, which the cancer cells can't use because you need a good mitochondria to burn those things. So that's the solution to the cancer problem. You lower the blood sugar, you target with drugs the glutamine, and you transition the body over to nutritional ketosis.
Okay.
And that
Okay.
marginalizes the tumor. So it's not one. You have to realize you have two fuels driving the dysregulated growth: the glucose and the amino acid glutamine, which is fermented in the mitochondria. It's not oxidized, it's fermented. And that's our new findings. So in order to manage the disease, you got to shut off the energy. And without energy, these cells can't grow. They can't manage the pumps and they die.
And
Not
your
that
and
not
for
that
anybody,
complicated.
right? And for anybody trying to understand this, your theory is that cancer cells generate energy through glucose, in large part once they're generating it. Now,
and glutamine and glutamine.
And okay, so Dr. Buns, what do you have to say about that?
Yeah. Well, that is very correct. Again, the metabolic profile of cancer cells has been studied for many years. We have a lot of insights as to sort of how that, how those cells behave. Let me tell you a little sort of anecdote, a little story about a really interesting experiment that was done a number of years ago that I think might perhaps shed some light on our discussion and might further give you a little bit of a sense of why, how does genetics figure into all of this? Again, with talking about the abnormal sort of mitochondrial functions, in cancers. I sort of agree with all of that. Cancer cells do have very aberrant metabolism. So one of the story I want to tell has to do with a cancer gene that's near and dear to my heart. It's called P53. It was discovered many years ago. We've been studying it for years, and we're still learning really fascinating things about it. This is a gene that's mutated at about half, give or take, of all human cancers. So it's very prevalent. It's certainly not in all of them, but in a good solid proportion. And so one of the ways that we've sought to learn about P53 is to basically alter that gene in mice. So, laboratory mice are a common tool for studying, as in a way of a surrogate for experiments that we can't obviously do on humans. So we actually, starting in the 1980s, developed technologies that have recently really come to the forefront to actually experimentally modify genes. So, we don't have to speculate as much about what genes are doing. We can actually go into cells and go into whole animals, modify a gene of interest in a very specific way down to the individual, DNA nucleotide. And we can then see what effect that mutation has on that animal. And so when P53 is mutated in mice, those mice become highly predisposed to develop a variety of tumors. Now, those tumors are very different from the tumors that arise in the context of P53 deficiency in people. And perhaps that's not surprising, since mice are very sort of anatomically different and they only live about 2 years. But anyway, so we can basically recreate in mice the causative effect of P53 loss. Now, now where the story comes into metabolism, a great friend and colleague of mine named Paul Huang, who's a prominent investigator down at the NIH in Bethesda, he did a really cool experiment. He was very much interested in studying P53 and its potential roles in regulating metabolism. So he took these P53, what we call knockout mice. Basically, the P53 gene had been inactivated in this particular strain of mice, and you could compare those mice with other mice that were completely genetically identical except for that one mutation. And so he did a really kind of cool experiment to kind of look at their metabolism, and he did a number of things, but maybe the most visually striking was he put them in a swimming pool. So the mice, how do you measure sort of the metabolism of mice and their energy level and their glucose consumption? So he had them in a very unique apparatus that they have at the NIH, where the mice are actually in a swimming pool with an engineered sort of current. And so the mice are sort of swimming towards a platform. And basically they're swimming against this current. And so the more the mice that have higher energy reserves, and the ones that have aerobic metabolism, which is a much more efficient means of generating glucose, and therefore and a much more efficient way of generating the ATP that mitochondria need to be efficient, those mice were able to sort of swim really well and reach the platform. The mice that had P53 mutations were all that you could see them even with an underwater camera. All the mice were sort of becoming exhausted. So they were unable to use glucose. So they were basically they were essentially fermenting glucose rather than they were using sort of what we call a glycolytic pathway to generate ATP, that sort of that molecule of energy from glucose. And so Paul, my friend and colleague, he took a closer look at this. So there's a clear metabolic defect in these mice with the P53 mutation. And when they took a closer look at it, it turns out of the many genes the P53 is known to regulate, one of them is a gene called SCO2, which is an integral component of the respiratory chain in mitochondria. So his study provided one of the very early links between a common genetic alteration and a metabolic defect that's ubiquitous in cancer. Now, as I mentioned, not all cancers have P53 mutations, so presumably there are also other mutations that can similarly, either directly or indirectly, affect these metabolic pathways.
Okay. Yeah.
Okay. Well, I'm familiar with that study as well. And P53 disrupts mitochondrial function and Li-Fraumeni and other conditions. And, you disrupt mitochondrial function, you're going to force cells to rely more heavily on fermentation in order to survive. So it's a mitochondrial metabolic disorder. And the tumors would be dependent on a fermentation metabolism for dysregulated growth. So you would then, to manage that, you would simply target the availability of the fermentable fuels in the body of the mouse, or the human, I should say. And I think Fred's right. You have to be cautious in how you look at certain animals with a 7-fold difference in basal metabolic rate compared to us. Right. But the issue, of course, here is that P53 mutations can occur in the germline, like Li-Fraumeni, or they can happen as secondary somatic mutations caused by a reactive oxygen species. So either way, you can have germline mutations in P53, or you could have somatic mutations in P53. Either way, you're going to disrupt the energy metabolism in the cells that would have those abnormalities. Therefore, they would be dependent. So that means the origin of the dysregulated growth is coming from a disruption of oxidative phosphorylation followed by a compensatory fermentation. So in order to manage those cancers, you would do the same thing you would do with any cancer, regardless of whether or not they have a P53 mutation, is that you would selectively reduce the sugar in the body and elevate ketones and target the glutamine. The glutamine is the second big fermentable fuel in these cancer cells, and they ferment it in the mitochondria through mitochondrial substrate level phosphorylation. So you're going to get those two fuels. If you take one away, you'll slow it down. If you take the other away, you slow it down. But to get rid of it, you really got to hit those two fermentable fuels simultaneously while the body is in nutritional ketosis. So because your normal cells will get really, really healthy burning fatty acids and ketones, but the tumor cells are marginalized.
So yes, Professor, would you agree? Just to sort of not to talk over you, but so would you agree though in those models and both in the mice and in individuals with Li-Fraumeni syndrome that the P53 mutation is the underlying cause of the metabolic dysfunction? So if we're talking a little bit about sort of what's the chicken and what's the egg?
Yeah. Okay. That's extremely important. Extremely important. So the question is, are they primary or secondary risk factors?
Is what now?
And whether you have a germline mutation as being a mutation linked to a neoplastic condition,
and a germline is just hereditary.
Yeah,
right?
yeah, hereditary.
Yes.
So that would be true if the Li-Fraumeni mutation were 100% penetrant. No known germline cancer that we have found is 100% penetrant, like Huntington's disease. You get Huntington's disease, everybody that has a mutation in the protein Huntington gets the disease. Li-Fraumeni is one of the highest penetrance risk factors that we know of genetically. It's about 85%. That means 15% of the people who have that mutation don't, maybe for a tumor suppressor genes or something like this. The primary cause is damage to oxidative phosphorylation. That's the
Well, I
primary
mean, the
cause.
reason again, the tumorigenesis is a time dependent process, right? So, those, again, there's this sort of the risk factor, and also people, this sort of that stochastic, sort of factor as to when people will lose that additional allele of p53, right? So in other words, so Seyfried syndrome for our listeners is basically a condition that's caused by a mutation in every cell in our body in the p53 gene. So normally we have two healthy copies of p53, one we get from the paternal side, one from our mothers. In some cases, if we inherit one defective copy of p53, that leaves ourselves with only a single functional copy. So we, in many ways, we're sort of, we can develop normally, but these patients are at extremely high risk of cancer. Now, why don't all of them develop cancer? Well, they develop cancer at different times in their lives. So it has to do a little bit with the luck of the draw when that initial founding cell of the cancer loses that remaining wild type p53 allele, and the timing of that can vary. So not everybody gets to age 45 with Seyfried syndrome and gets cancer. There's a range, and some people are sort of that range will be outside of their normal lifespan, so they won't get cancers. In some respects, Professor, the penetrance of Seyfried syndrome is actually much closer to 100%. And let me tell you why. So among the really interesting phenotypes of p53 mutations, in other words, what are the characteristics of people that have a mutant p53 in their germline? So people with who suffer from Seyfried syndrome also have a metabolic phenotype. It's really interesting, and I've talked to physicians that actually take care of these families and sort of keep track of, they kind of monitor them for tumors because they're at such high risk. One of the really memorable things that one person told me once is he can look down the hall. So often, families that are afflicted with this disorder will bring, they're bringing the whole family in for a consultation, the kids, everybody. You can actually see from down the hall which individuals are affected by Seyfried. So again, it's a monogenic disorder. So not everybody will. So about half the offspring from a normal individual and an individual with Seyfried syndrome, only half the kids will carry that trait. If you look down the hall, you can see just by their sort of body mass index, you can sort of tell which are the Seyfried mutation carriers. So it's so it's really to my mind, it really creates a vivid illustration of the underlying role of genetics in this type of metabolic dysfunction.
Well, I mean, I'm not opposed to that at all. I mean, when we know that mutation affects the electron transport chain, what is intriguing to me is that you would have a few, and you're right, it's very high penetrance. I'm not arguing with you. In fact, it is the highest of any germline that I know of. Seyfried is the highest penetrance. But again, it's not 100%. And every one of the people who do eventually get a tumor, whether it's a brain tumor, ovarian tumor, they have a range of different kinds of tumors in the body of those folks. Every one of them is fermenting. And don't forget, you need oncogene turn on. When you have damage to oxidative phosphorylation, oncogenes must turn on, the HIF1 alpha, the MYC, and all these. They are facilitators of bringing the fermentable fuels into those cells in order to allow them to be shift to the fermentation. So you have to know when the genes are playing a role. As I said, if you put a new mitochondria, the oncogenes turn off because there's no need to ferment, and they are the facilitators of fermentation. Yeah, Seyfried, all the tumors that we have looked at, they're all basically fermenting because the mutation itself damages oxidative phosphorylation. So the linkage is very tight. But all the others like BRCA1 and retinoblastoma, they also damage oxidative phosphorylation. The penetrance is lower than Seyfried for sure. But every one of the germline mutations that I have looked at has damage to oxidative phosphorylation with compensatory fermentation. Now, if you damage oxidative phosphorylation and do not get compensatory fermentation, that usually causes death because you're going to stop the energy. You got to have energy either from fermentation or oxphos. If you shut down oxphos too quickly, cells will die. They won't form tumor. So you have to have the upregulation of the compensatory fermentation, which is driven by oncogenes. But these are facilitators. They're the effects, not the cause.
Yeah, well, so let me actually address. There's a point that you raised earlier about. I believe you mentioned these findings that you can actually find cancer, tumor suppressor and oncogene mutations in normal tissues, right? So how could that be? It's a very important observation. I think it's sort of worth addressing. And you're absolutely right. They do find mutations that even in some cases have clonally expanded in, for example, skin tissue. They found them in the crypts in the colon. These are very common findings, actually many of which have been made by Michael Stratton's group, who I think would be very much in the camp that the mutations cause cancer. And so what a key factor of sort of genes as a cause of cancer is that one genetic alteration is not sufficient. So that's sort of a key, I think takeaway for this is that it's, for example, if you look at even tumors that arise in leaf brown Mayne patients, those tumors will have that germline p53 mutation with loss of the wild type allele, but they're going to have other mutations as well, other oncogenes that contribute to tumor growth. And as you described, many of those genetic changes also affect metabolism. For, the RAS signaling pathway, which is a highly recurrent, really recurrently mutated signaling pathway in a whole range of cancers, is known to be very important, for example, in glucose transport. So again, the underlying genetic alterations really fuel a lot of these sort of metabolic abnormalities that are universally seen across tumors.
Yeah, well, we know the RAS mutation damages oxidative phosphorylation. Pen Wang showed that a while ago, that mutation. You don't get cancer with healthy mitochondria. If you want to prevent cancer, you got to keep your mitochondria healthy. All cancer cells that we know of have abnormalities in the number, structure, and function of mitochondria. That's the commonality of all cancer. They have different genetic mutations for sure, but that's a downstream effect of the RAS production.
Yeah,
Every cell.
So what we're saying again with the P50, that's really the upstream cause, right? So if people are born with the P53 mutation or a BRCA mutation for that matter, we know that they're at elevated risk for cancer, right? So that's why we do BRCA screening. Again, to sort of know which people and families to follow. You can't get that information by looking at their mitochondria.
No.
That's manifest later.
Yeah, no, that's not what I'm talking about. The mutation BRCA1 damages oxidative phosphorylation. That's what I'm saying. So we have to recognize what is primary cause of dysregulated cell growth versus secondary cause of dysregulated cell growth. If we have 50% of the women that have BRCA mutation have breast cancer, there's 50% of the women that have BRCA mutation that don't have breast cancer, and every woman that has the breast tumor has damage to mitochondria. That means the mitochondrial damage is the origin of the dysregulated growth, because we can have the mutation without the dysregulated growth, but we can never have the dysregulated growth without the damage to the mitochondria, insufficient oxidative phosphorylation. So mutations are neither necessary nor sufficient to lead to dysregulated cell growth, but mitochondrial inefficiency is necessary and sufficient with a compensatory fermentation. So I think we always have to put into
mutation, though, is a known risk factor, right?
Well, you have risk factors for sure. But is it a primary risk factor or a secondary risk factor?
I mean, it has to be a primary risk factor because you can identify who's at risk by genetic sequencing.
Yes, but they don't all get cancer.
Right, but you can know who's at risk, right? Again, as we've discussed.
No, I'm not saying you can't.
Does every person who has mitochondrial disruption get cancer? Does that? No,
that's you have to have the compensatory fermentation. You have to. If you have a damage to the mitochondria, you'll kill the cell quick. But the question is, getting back to primary and secondary, because I think this is so important when we're looking at origins. Is the mutation a primary cause or a secondary cause? I look at it, if it's a primary cause, every time you have that mutation, you get the tumor, regardless of whether it's a breast tumor, colon tumor, whatever it is. It's 100% penetrant. There's no ambiguity. But if a person carries the mutation and never develops the tumor in their lifetime, then that was not a primary risk factor. It was a secondary risk factor.
Well, but then how do you explain the increased risk, on a population basis of people that carry a BRCA1 mutation?
Yeah, but there's a lot of women that
why
have
are they getting
BRCA.
more? Yeah, but why is there more cancer in that population?
Because that mutation damages oxidative phosphorylation.
Right. So that's a cause and effect, right?
Yeah. But you cannot find a tumor without damage, insufficient oxidative phosphorylation. But you can find women with BRCA1 mutation that don't have cancer, or ever get cancer in their lifetime. So yes, it's a very significant risk factor, but it's a secondary risk factor. The key is the primary. What is the primary elicitor of dysregulated cell growth? And it is insufficient respiration with compensatory fermentation, which has been found in every known cancer that anyone has looked at. No one has ever seen tumors with normal oxidative phosphorylation. In vivo. In vitro, you see this because oxygen consumption is not an accurate marker for respiration in vitro, but in vivo it is. So I think you have to look at. I think it's important for people to look up the differences between primary and secondary risk factors. Smoking is a secondary risk factor. Obesity is a secondary risk factor. Insufficient inflammation, viral infections, papillomavirus, hepatitis. These are all secondary risk factors. They cause cancer only if they damage oxidative phosphorylation. So then you get dysregulated.
Yeah, so let me just say this concept of a primary and a secondary risk factor is really not a widely accepted sort of concept. So when we, in sort of maybe I would argue the mainstream, we talk about risk factors. There are sort of risk factors, but there are also underlying causes. And so those are two different things. So if you create a mouse that's going to get cancer later in his life, the underlying cause is that genetic alteration. And if you expose that mice to chemicals or do any number of other things, that mice or radiation, that mouse will likely get cancer at a younger age. So you can look at, again, in human populations, again, we know that these germline genetic mutations are profoundly increase our genetic risk. But other things can increase that risk also. So all of us have multiple sources of cancer risk. So when we talk about, all risk factors are, they have different magnitudes, but we don't really think of primary and secondary. The primary underlying cause are the cancer mutations that fundamentally change how cancer cells operate and how they function, including their metabolic defects. Again, this is a point we disagree on, but
I look at the nuclear cytoplasmic transfer experiments. If the neoplastic growth in a tumor is driven by genetic mutations in the nucleus, according to the somatic mutation theory, dysregulated cell growth is caused by 1, 2, dozen mutations, doesn't make any difference, drivers and whatever. And you take that nucleus and put it in a new cytoplasm, and you no longer get dysregulated cell growth. That says that those mutations are, whatever they were, no matter how many there were not the drivers of the dysregulated growth. If I take the normal nucleus and put it in a tumor cytoplasm, I get dysregulated growth. So clearly it's the cytoplasm and the mitochondria. And these experiments have been done over and over again in all kinds of different systems testing. They were not testing the somatic mutation theory, but the results from those experiments clearly tell us that the mutations in the nucleus of these tumor cells are not the drivers of dysregulated cell growth. I mean, it's hard to reject those findings. You can say, well, they're primary causes. Okay, if that's the primary cause, how come when I put that nucleus in a new cytoplasm, I don't get dysregulated growth? And if I take a normal nucleus, healthy, and put it in a cancer cytoplasm, I get dysregulated cell growth. How do you explain that? That's not explainable on the somatic mutation theory, no matter how you cut and look at it. You got to think. And what troubles me is that a lot of folks ignore those nuclear mitochondrial cytoplasmic transfer experiments that clearly show that this. And the only person that really understood that was the late Ruth Sager at Harvard. She said these nuclear transfer experiments support Otto Warburg's theory of cancer because you're not getting the dysregulated growth. When you put a tumor nucleus when you put a tumor nucleus into a normal cytoplasm. So why you don't get dysregulated growth when it has all the driver genes and the broken chromosomes and the somatic mutations? Why that doesn't cause the dysregulated cell growth? That's the big question. And the bigger question is, how come nobody discusses these kinds of experiments? This is my conundrum. People were saying, well, I didn't know about those experiments. Well, I think everybody needs to know those experiments and look at them and say, how do you explain this phenomenon of cancer dysregulated growth based on these nuclear cytoplasm transfer experiments? They're not consistent with this somatic mutation theory. So I want to just say that, and I think people need to look at all the evidence. I don't look at it any different from the geocentric heliocentric theory of the solar system. For 1,800 years, we were using epicycles and deference to try to explain how planets and celestial bodies revolved around an immovable Earth. Okay, it took 1,800 years before Copernicus said, maybe we got it all wrong. Maybe the sun is the center and the planets are all revolving around the sun. And then Kepler came in with the helical orbits, and Galileo with the telescope, and all of a sudden everybody says, the Earth revolves around the sun. I look at the somatic mutation theory and the mitochondrial metabolic theory as at exactly the same position that we have today. Once we realize that cancer is a mitochondrial metabolic disorder, the solution to the problem will be quite apparent. And when you take people that have cancer and remove the fuels that drive the dysregulated cell growth, then you start to get management of the disorder, and people will start living longer at a healthier overall and quality of life that is progression-free, and overall survival will increase dramatically. And we're beginning to see this now in all these stage 4 cancer people who are shifting to metabolic therapy. So the bottom line is, I think as long as we consider that cancer is a genetic disease, we're not going to drop those death rates in any significant way for the foreseeable future. Once you realize that it's a mitochondrial metabolic disease, I think we can drop the death rate by 50% in 5 to 10 years, depending on how you go after it. You're going to see that needle drop significantly. But if we're chasing genes and we're doing these genomic mega maps and all this kind of stuff, and all these people are dying from cancer, something has to change here. And I think you got to look at the nuclear transfer mitochondria. You have to look at all the data, driver genes that don't cause neoplastic growth, nuclear transfer experiments. And then you have to say to yourself, maybe we got it all wrong. Maybe we're not right. And that's what the field found when you put the sun in the center of the solar system. Everything made sense. You didn't need epicycles any longer.
Okay.
So
So
that's my view. And I think I'm right. And I think we're going to drop this death rate very, very big once people come to realize that they can't live without a fermentation metabolism. And that's not difficult to target. Targeting fermentation is a hell of a lot easier than targeting a bunch of genes which may or may not lead to overall outcome.
Now I only have time for one more question. So I want to get this one out, and it will give you the opportunity, I believe, to respond, Dr. Buns, if that's what you're hoping to do, which I imagine it is. Now, this will go back into treatment and management of cancer. Okay. We have all these different treatments right now that sort of on a spectrum are helpful. Some aren't. It just depends on the person. A lot of times, I've personally witnessed somebody in my life go through chemotherapy they were diagnosed and then went through chemotherapy, and 4 to 5 weeks later they died. And so it really took them out. Now that happens a lot. And also chemotherapy helps a lot of people too. So I'm not saying it's all bad, it's all good. Some of the treatments for cancer, you have chemotherapy, hormone therapy, immunotherapy, radiation therapy, stem cell therapy. There's a bunch of others. And speaking with you, Dr. or Professor Seyfried, you have talked to me before about other solutions to manage, or more or less to manage cancer, kill cancer cells. I would imagine that your take on some of these more traditional cancer treatments is that you probably would say they are not as beneficial for people to use today, based on everything you just said. But I wanted to ask you, Dr. Buns, if you think that if there's one particular cancer treatment that you have seen to be the most effective for treating cancer, and why. And then I would ask you the same thing, Dr. Seyfried, but you go ahead, Dr. Buns, first.
Yeah. So, these are really exciting times for cancer research, for a whole number of reasons. But, and maybe I'll sort of using your sort of, looking at the different theories for how the heavenly bodies move in the universe. I think so. The heliocentric theory, in my view, is really, I think it's a nice analogy for what we're seeing really with the role of genes in cancer. So in a sense, the cancer gene theory in biomedical research is sort of, in my view, sort of the equivalent of the heliocentric theory. And why is that? So I guess what we can ask: what's the strength of the heliocentric theory? Is it just one group of people arguing with another group? And I guess there still are people today, right? Who think there's a flat Earth, or the sun's going around the earth, or whatnot. But the really wonderful thing about the heliocentric theory and other theories like it is that is their power for us to understand the universe, and for the power for us to make certain predictions. Right? If we know how the heavenly bodies move, it helps us understand tides. We can predict the next solar eclipse, down to the minute, down exactly to where it's going to happen. So all of these types of things, we can use our knowledge of planets to sort of launch a probe into the heavens, and have it, years later, sort of fly, within a couple of kilometers of Pluto. And this is because we know what the heliocentric theory empowers us to kind of to do all these amazing things. The cancer gene theory is really like that in a lot of ways. So talking about some of the therapies that have sort of come up in recent years, and some of those that are in the pipeline, some of the targeted therapies are really profoundly changing the outlook of people with certain kinds of cancers. Now, certainly there's some cancers we haven't been able to hit very well, but there are a number of leukemias that have famously and very dramatically responded to targeted therapeutics. Now, what are those therapeutics targeting? They're actually targeting the products of cancer-associated mutations. So, for example, the classic example is Gleevec, which is really the first widely used targeted therapeutic against a specific genetic alteration in a type of leukemia called CML, or chronic myelogenous leukemia. So we can predict who's going to respond to Gleevec by sequencing by molecular characterization of their tumors. And similarly for EGFR inhibitors in lung cancers, and on and on. So the list, fortunately, is growing for very targeted therapeutics that we can use to target certain genetic alterations. In addition, now, a particular type of therapy I'm really very bullish on, and I think we're going to continue to see really great strides in this area, is our immune-based therapies. So basically taking advantage of the different ways that the immune system recognizes normal cells versus cancer cells. And we know one very important aspect of what makes tumor cells immunogenic is really their mutational load, for example. So all those different genetic alterations that occur in the cancer cell, cause the production of these abnormal proteins, which then can in turn be recognized by the immune system. But cancers also, as a result of genetic alterations like P53, will sort of downregulate the presentation of those antigens. So I think we're learning now of ways of activating T cells against specific cancer-associated genetic alterations. And I think that's really where the next generation of really powerful therapeutics is going to come from. And so we'll kind of get away from using what these treatments are essentially poisons to really using our knowledge of cancer genetics to devise very specific therapies that can really target those specific alterations and, leaving the rest of our tissues, healthy and able to sort of, continue to grow normally. So I think that's something that we have to look forward to in the decades ahead. And well, hopefully the years ahead. I don't think it'll even really be decades.
Okay, awesome. And Professor Seyfried, I'm going to ask you the same. And I also have to preface this with, I just got an alert that I have 10% left on my camera.
Yeah. Well,
so keep it as concise as you could, please.
Well, I think the immunotherapies are based on the somatic mutation theory. We have 1,700 people a day dying from cancer. Those therapies have done nothing to drop the death rate. As a matter of fact, they've looked at all these genes over the last 92 genes, and they improve overall survival by 2.4 months. I mean, let's be honest. These poor people are suffering immensely from a misunderstanding of what the nature is. We have what we call hyperprogressive diseases. You give immunotherapies, yes, some of those targets are on normal cells. You blow out somebody's liver or kidney as the result of trying to help them. What is going on with that? Why would you treat any member of our species with any kind of a therapy that has a remote possibility of hurting them, or worse yet, killing them? I mean, this CAR T immunotherapy, Keytruda, Opdivo, this is based on an incorrect theory. We're not going to drop the death rate anytime soon, as long as you consider this is a genetic disease. Fred, you're just pushing. We're not going to drop the death rate, my friend. That stuff ain't going to help. There's massive evidence to say it's not dropping the death rate. It's killing people. And people are making a huge profit on drugs that don't work. Yes, there's always the blind squirrel that finds a nut, and you get somebody that really responds really well to an immunotherapy.
Clinical trials, right?
No, trials are killing these.
That's what trials
They're not working. They're not improving overall survival in any significant way. You know that. You know the literature. So, look at the survival. Why do we get all these people dead? You want to know if something works, you drop the death rate. You drop it big time. You choose metabolic therapy, you're going to drop that death rate. But God forbid anybody tries it. What happens if we do metabolic therapy and drop the death rate by 50% in 10 years? Are we saying, we have the wrong theory here? Let's go back to the gene theory.
We should be targeting tumor metabolism. And actually, people are working on that.
Well, actually, that's an important point, Fred. Right? You know, the immunotherapies work better when you take away glucose and glutamine and transition the body to the... Now, let me tell you something. If you shrink the tumor down and you got a very small tumor, the surviving cells may have something in common. And there's where an immunotherapy could come in and be a very powerful tool. But it's at the end, not the beginning. So because now you take advantage. We have all the tools needed to manage cancer effectively. We're just not using them in the appropriate way at this time and place.
Okay. And with that, I think what I'm going to say is I'm going to direct everybody to go to both of your different pieces of work, your literature that you have created on these topics. Very, very important work that both of you have done. I think that if we wanted to, we could do a follow-up on this with some additional questions if that's something that we wanted to do. I'd ask, everybody watching to let me know your thoughts on that if we ought to. But where can people find your work first, Dr. Buns?
Yeah. So my textbook is available on Amazon and a bookseller near you. So I'd invite that's a great introduction, I think, to the topic, where I talk very broadly about cancer etiology, all the way through therapy. So I think that'll hopefully be useful for people that are trying to get a little bit their feet wet in this. Yeah, and then again we publish in very focused areas of cancer research, and I really invite people to really look at any of these studies that we've been involved with over the years. So I think it's really again I the one thing I will agree with is that too many people are dying of cancer, but it's really not for lack of effort, and I think we're rapidly entering, I think, a new era where we're really going to see some really real improvement in some of the most refractory types of cancers.
Awesome. Yeah, that's the hope. That's what we're going for. And same with you, Professor Seyfried. Where can people find your work, please?
Well, I published a book also in 2012, which is selling more now than it's ever sold. But I also publish all open access. We're encouraged here at Boston College to publish open access so people can read. There's no paywalls that hide the stuff. So guys, anybody who wants to look at this, just put my name in and you'll get the open access publications. Because the change will come from the demand of the people. Once the people come to understand what's going on, they're going to make the change. I don't think change is going to come from the top. It's going to come from the bottom. And when you get more and more people educated to know what the nature of cancer is and how to effectively manage it in that we write about, then you're going to start to see the change. Until that happens, I don't see much, but I agree with Fred. There's going to be a massive paradigm shift coming very, very soon. And the cancer patients are going to benefit from it immensely.